I'd like to make this informal, so as I go through this talk, if you have any questions, please let me know and we'll discuss it as I go along. As I said, I'm going to go through some of these slides, assuming we can get the slides up pretty quickly, so please bear with me. While we're pulling this up, how many of you have gotten to know about genetically modified organisms and biotechnology and food? And how many of you have been asked questions about it in the course of practice? Good. So it's a large topic, and it's not the kind of topic that most of us have the time or the ambition to become totally familiar with. There's a lot of DNA chemistry involved in it, a lot of basic experimentation involved in it, and it's a topic that is, I think, ripe for misunderstanding. So I put this talk together first a couple of years ago for the International Congress of Nutrition that was held in Granada. It's not the same talk that I'm going to give you now, but it's what made me decide that this was a topic that probably was worth talking about, and so I worked to try to refine it more towards pediatrics compared to what I did a couple of years ago, and that's what I'm going to do this morning. So once we get this up, I'm going to provide some background on the reasons why this is relevant to us. I'm going to go into the history of food modification over the centuries. I will talk about biotechnology and how it's tested and its effects on the environment, on humans, and on animals. And then I'm going to conclude with some focused remarks about biotechnology and pediatrics. Many of you probably also saw the commentary that came out from the WHO, a group in the WHO, about Roundup, glyphosate. Is that true? Have any of you seen that in the paper where they classified Roundup as a probable carcinogen? It was swamped in the last few days when they also classified meat as a probable carcinogen. And so I think their influence is somewhat limited. They have now considered something like 949 different substances, methods, and so on, everything from working the night shift to drinking coffee to... Are we up? Oh, yeah. That's okay. There we go. And of those 949, they've classified 948 as carcinogens. Only one of those things has not been classified as a carcinogen. So at the end of this talk, should I get to give this talk, I'm going to touch on that because it is relatively fresh, and while Roundup is not a genetically modified product, it's a chemical, nevertheless, 80% of it is used on genetically modified products, and so it has a lot of relevance to this topic. All right, well, we were going to go through this at 50 miles an hour. We're going to go through it at 75 miles an hour now. So take a look at the pictures on this slide. I'm going to come back to these pictures in just a minute. I have no disclosures to make, and it seems clear, as you can see from this picture, the cover of National Geographic's just this past March, that we seem to be entering a period where there's a war on science. Climate change, evolution, vaccinations, and genetically modified foods are all areas in which there is extremely solid, broad consensus. More than 85% of the scientific community is in agreement that these things are real, valuable, and important for us to deal with. And so I think that is the environment in which we're working today, and all of us in pediatrics, of course, probably have experienced this most with vaccinations. These are some questions that often come up about GMOs, and I'm not going to go through them all, but we will touch on just about all of these questions in the course of the discussion. And so, as I said, I'm going to provide some background on infant and child mortality, going through then a history of improving foods, then biotechnology, and finally summarize by talking a bit about Roundup or glyphosate. Why does this affect us as pediatricians? Many of you told me that you've been asked questions about this directly in the course of your practice. There is a lot of emotion around this topic, an awful lot of misinformation, misleading information, and particularly in the blogosphere, we see things like this that then come right back to us in the office when parents read these things and wonder what they should do. So if in any way you work in nutrition, and every pediatrician works in the field of nutrition, then you really do need to know something about GMOs. We know that we use biotechnology every day. Half the vaccines that we use today are products of modern genetic engineering. Insulin, 99%, 100% of the insulin used today is genetically modified, biotech insulin. Albumin, all of the new biological drugs are products of genetic engineering. But even more than that, anybody who is alive today encounters products of biotechnology every day. We had a lovely dinner last night with some wine. That wine, the yeast used to make that wine is a product of biotechnology. 90% of the cheese is produced using enzymes that are created through biotechnology. And you can read through the rest of this list and recognize that there's no aspect of life today as we know it that doesn't embrace biotechnology. And in fact, if we were to turn the clock back and stop using biotechnology, it would be an awful lot like giving up your cell phone. It's just not something that you can do and do it easily. So how about biotechnology, nutrition, and health? I want to turn to the WHO 2015 Millennium Goal, which was to reduce deaths in children under 5 worldwide by half. And the good news is that that goal has largely been achieved. 16,000 fewer children die each day compared to 1995. However, there are some parts of the world where this isn't true, and some categories of infants and children where this isn't true. Sub-Saharan Africa is one of those, Southeast Asia is another, and throughout the world, the proportion of deaths that occur during the first month after birth is actually increasing. And there's a direct relationship between these deaths and family security. And this has been well described now in this schematic or format that was put together by UNICEF back in 1990. And what this says is that underpinning infant nutrition and hence infant mortality is household access to adequate quality and quantity of resources, including land, education, employment, income, and technology. And biotechnology has had a direct effect on this, and I'm going to talk about that as we get into the talk. So households with these resources have good food security, and those without have food insecurity. Food insecurity leads to inadequate dietary intake, and inadequate dietary intake in infancy and childhood leads to malnutrition. And the risk of dying if you're malnourished and have pneumonia is over 10 times the risk if you're well nourished. And the same goes for diarrhea and other acute infectious illnesses in infancy and childhood. This is brought out in this table, and you can see I just highlighted 2 of the categories, mother's education and her occupation. And there's a 5 to 8-fold increased risk for mortality in neonatal, postnatal, infant mortality, child mortality, if the mother is illiterate, compared to whether she's had a secondary or higher level of education. And the same goes for if she's a professional versus a laborer. So let's leave that for the time being and move into the history of genetic change in agriculture. Remember the very first slide that had the title of my talk on it, and there was a picture in the lower left and the lower right. On the lower left of that was a picture of primitive corn called teosinte. This is a plant that still grows today. You can find it in Mexico. The kernel on this plant looks like this. It's dry, it's dark, it's hard, it's bitter, it looks nothing like corn. But through multiple cross breedings, starting about 10,000 years ago, we developed modern corn. So modern corn never existed in nature. It's a product of genetic engineering, and the genetic engineering occurred through crossbreeding. And while I said that this started 10,000 years ago, and there's evidence of this in ancient drawings, and you might say, well, 10,000 years has allowed for it to evolve gradually, and therefore any issues that might arise from that kind of genetic engineering have been worked out over time. Well, think about the corn that you eat today. It's nothing like the corn that I ate as a kid. It's much sweeter, it's crisper, it stays fresher longer, and all of that has come about in the last 20 years. In fact, almost nothing that we eat today, all of the pets that we have in our homes today are products of genetic engineering. Dogs, chickens, pigs, on and on. When it comes to vegetables specifically, these are examples of some of the other vegetables that we eat and what they look like before they were crossbred or modified genetically. From lettuce to root vegetables to cruciferous vegetables, none of them are found in nature. And some of these crops didn't even have progenitors in nature that look like them. Wheat is an example. Wheat is a cross breeding between spelt and goat grass, and several other cross breeds to produce modern bred wheat. And the wheat that we had last night in that lovely Italian restaurant was produced through a process called chemical mutagenesis and radiation breeding. And if that doesn't sound scary, I don't know what does. And I'm going to come back to that in just a minute. So why then is it kind of false to talk about genetically modified organisms as a separate class? Why is it that anything that we do to modify an organism genetically is relevant and really in the same category? And of course that's because we have a single code for life. It's the genetic code. It's based on DNA being translated to RNA and transcribed to protein and so on. And this code is the same for all living forms. And there actually is significant homology between all living forms and humans. For example, going backwards on this slide, we're about 98% similar in our expressed genes to a chimp. To a mouse, 92%, yeasts, 26%, and we know this because we use these organisms as model systems to better understand human biology. And so taking a gene from one of these organisms and implanting it into a different organism, like a fruit or a vegetable, is no more dangerous or relevant than taking from the same plant and modifying it with another similar plant. It's all DNA. And here I think is one of the most relevant slides. Traditional plant breeding, whether it's crossbreeding or chemical mutagenesis or specific use of biotechnology, traditional plant breeding takes a trait that we want to put into a vegetable, let's say, corn, we want to make it sweeter. and we find a sweet type of corn that doesn't have all of the traits that we want when we breed this new corn plant. It doesn't stand up very well in the field. It doesn't yield very much corn when you plant it. And so multiple crossbreedings are carried out in order to get this new sweeter corn. In the process of doing that, multiple genes are transferred from the starting plant to the finished plant. We don't know what those genes are. We do know that embedded somewhere within those genes is a gene for sweetness, but a lot of other genes are carried along in that process. Biotechnology allows us to be extremely precise. Only the gene for sweetness is going to be moved from the target plant to the new plant. None of those other irrelevant genes are going to be carried over. And so it allows a level of precision and therefore potentially a level of safety that actually doesn't exist in conventional breeding. The issue about time and how long it takes to do this has also been misstated. On average, it takes 8 to 10 years to complete this process for GMOs using modern biotechnology. You have to spend anywhere from 2 to 4 years discovering the gene for the trait that you want, then putting it into plants, so proof of concept, planting it and seeing how it survives in the field, and then further development and safety testing, all of which take between 6 and 10 years. So the idea that you pull a gene out, you put a gene in, you put it into the field, and bang, it's in the grocer's fruit and vegetable aisle 6 months or a year later is simply not the case. So these are how crops have been genetically modified. Traditional breeding, I've talked about that. I mentioned this chemical radiation, chemical mutagenesis and radiation. Some 3,000 plants, from the wheat that's used now in the best pasta, to organic rice in California, to ruby red grapefruit, to 2,000 some odd other commonly consumed vegetables are produced this way. There's significant genetic disruption as a consequence of this technique. They have been safe so far. Nobody has brought up issues of safety, but they're not tested at all, any more than conventional plants are tested. And then we have modern techniques like RNA interference and transgenics, using RNA to knock down a gene, or transgenics moving one gene, moving a single gene or two from one organism to another. And in this case, extensive testing goes on. So I want to go through some of that testing with you now. First is testing the gene. The source of the genes are carefully examined, their character, the molecular characterization of the gene is taken out, and then when the genes are inserted, copy number, gene integrity, and so on are carefully assessed. The protein produced from this gene is examined across a vast data bank now of all known proteins expressed in life. It's also cross-matched against large allergen data banks that have all known allergens and their molecular composition. In addition, these plants are tested for food and feed composition, animal performance, key anti-nutrients, and so on. And they're tested in environmental studies to make sure that they have no adverse effects on the environment. And this is part of the reason that it takes 6 to 10 years to make one of these come to market. We now have very sophisticated ways to assess whether or not what's produced by a genetically modified plant is the same as its cousin from a conventionally bred plant. And this is a paper from last year which used liquid chromatography to examine mass spectroscopy to look at all metabolic components of tomato plants. And they took 6 different tomato plants, isolated 1,000 compounds in those plants, and the only thing that differed between the conventional plant and the new plant was the protein produced to increase sweetness in the tomato plant. Nothing else differed. So these are identical, and it's because of this testing, this extensive and fairly elegant testing, that international independent agencies across the world, some 240 now across the globe, have asserted, confirmed that modern biotechnology is safe and at least as safe as conventional and potentially even safer. And I've given you 10 examples here. And in the U.S., the AMA, American Association for Advancement of Science, National Academy of Sciences, and so on, all have affirmed the safety of these. And they've done that based on a literature that is not a literature produced by Monsanto and other big agribusiness. In fact, the majority of the studies that have been evaluated are studies that have been independently funded and have confirmed the safety of these plants. And here it's been laid out from the genera database of all studies that have been done on biotechnology in plants and animals over the last 20 years, and there's now about 2,500 of these studies. And these studies are both short and long-term in multiple organisms over multiple generations. And they cross the globe. They're not just being done in the United States, but everywhere throughout the world. And as a result of these findings and these studies, biotechnology has been adopted throughout the world. And this is the growth of biotech planting across the globe over the last 20 years. And now over 400 million acres have been planted. You can see the slope of these lines, and you'll notice that they are being adopted in developing countries at an even greater rate than they are in industrialized countries. And the reason for this is that in those countries, 90% of the farmers who adopt GMO technology are small farmers who have farms of less than 20 acres and who are at middle or poor income levels. 27 countries in 2013 were growing biotech crops, and others across the globe have been significant importers. In the U.S., 5 major staple crops now are almost all produced via biotechnology. Soybeans, cotton, corn, canola, and sugar beets are the 5 top crops. And as of 2015, better than 80% of all these crops are modern biotech crops. So why have they been adopted to this degree across the globe in small countries, in developed countries, and in developing countries? Well, because they produce seeds that have insect resistance, that are tolerant to herbicides, that are resistant to viruses, and that can potentially resist drought and salty conditions in the soil. And you can actually stack these traits in a single plant so that you get the benefit of all of this in a single plant. This is a farmer in the Philippines. I actually met this guy. I was invited by the Philippine Pediatric Society about 15 years ago to address their annual meeting. It's commonly said that biotechnology forces small farmers out of business. This is a tool of big agriculture. I mentioned to you that 90% of those who use biotechnology in the developing world are small farmers. And the reason that they do that is because it raises their income by about 34%. And in this particular case, for this farmer, it was the planting of BT corn. BT corn resists the corn weevil. So that's a pest that infests corn. It causes the corn to rot on the plant. And when it rots on the plant, it becomes super infected by fungus. The fungus produces mycotoxins, and so if you're a farmer working in your field and you're exposed to these mycotoxins, you're at increased risk for a whole variety of terrible things happening, including liver cancer in this young woman and meningomyelocele. And through the use of BT corn, This particular farmer and others like him have been able to reduce exposure to these mycotoxins by better than 90%. Tolerance to herbicides, and we're going to come back to this. This is glyphosate Roundup. Why does this work? Well, plants make their own aromatic amino acids. We have to eat them. We don't have the metabolic pathways to make aromatic amino acids. The specific pathway that plants use relies on an enzyme called EPSPS. Glyphosate can bind to that enzyme, prevent the production of aromatic amino acids, and the plant dies. So if you're like me and you don't like weeds in your garden, you spray some Roundup, the weeds die because they can't make proteins because they're prevented from producing aromatic amino acids. But if you could make some plants immune to the binding of glyphosate to this specific enzyme, then you could spray it without worrying about what you're spraying it on, and you could kill the weeds but not the resistant fruits and vegetables that are being sprayed. So it's a way to kill weeds between corn plants or any other type of plant without tilling. And so you enhance the quality of the soil because you can employ no-till farming by spraying glyphosate on those farms. And while you are still spraying an herbicide, you'll see that the kind of herbicides that we are spraying or farmers are spraying today are much, much less toxic to humans and to the environment. And in fact, there's no toxicity of this to humans because we don't have that enzyme pathway in our metabolic processes. Viral resistance is another important trait. In Hawaii, papaya is a major cash crop. And in Hawaii, 10 years ago, the papaya ring spot virus had begun to wipe out the crop to the point where 90% of the crop had been wiped out. Through genetic engineering, plants were produced that are resistant to this virus, and today the papaya industry, papaya planting in Hawaii is flourishing. And that's true for squash, potatoes, multiple other crops. And drought. And here we're getting now close to home. We think about Africa because major droughts have occurred there and in Southeast Asia. And in developing countries, they are heavily reliant on rainfall to irrigate their croplands. They don't have the irrigation infrastructure present in industrialized countries. And so they get into a vicious cycle of lack of investment, instability in the environment, and drought. And these droughts not only bring dryness and lack of water, but they bring other pests as well with them. And in 2005, Niger underwent a severe drought. 62% of the population became severely malnourished as a result of this drought. These are pictures from California. So this is happening very close to home now. Climate change is a reality. We need to be prepared for it. And using genetic engineering, we can have the corn on the right that is bred through genetic engineering to be drought and salt tolerant, or we can continue to plant the traditional plant that we have on the left, which has been grown under drought conditions, and if you look carefully, you can see that those are not healthy ears of corn. The world population is growing. The environment is changing. The growth of the world population is pretty dramatic. It'll go up by about 50% over the next 20 to 30 years, and the need for food will rise significantly. These are some of the increases in staple crops that will be required over the next 20 to 30 years. And yet at the same time, the available land for planting is continually shrinking. It's shrinking because of cities that are forming. Land is being used for housing, and it's shrinking because arable land is is not available as a result of drought and climate change and other factors. So if we're going to produce the amount of crops necessary on the left side of this slide, we have to be able to embrace a number of different aspects employed in farming today. And these are the yields, the change in yields over time since the mid-19th century to today. And you can see that beginning around 1935 to 1955, there was an upward tick. That's when fertilizers and so on began being used. And then there was a dramatic uptick starting around 1965, and that's when the Green Revolution took place. This was championed by Norman Borlaug, who won a Nobel Prize for it. And he talked about the need for specific targeted irrigation, specific targeted fertilization, rotation of crops, many of the things that are employed in farming today that have led to these kinds of yields. And biotech has contributed to a continued increase in yield over time. But in those places where they're not using biotech or modern farming techniques, yields are the lowest in the world. And unfortunately, sub-Saharan Africa, an area where infant and child mortality is rising, drought is highly prevalent, they are not using this technology, and these are the kind of yields that they're seeing. And here's a lesson, because this isn't true throughout Africa. In Malawi, for example, they were importing food between 2005 and 2007. When they adopted biotech plants and seeds, they were able to go from a deficit in 2005 to a surplus of food in 2007. It's about the most dramatic natural experiment that I've seen on the value of the use of biotechnology. One of the myths that's been on the blogosphere is that the use of biotechnology in another underdeveloped area, India, has led to mass suicides among farmers because they couldn't afford the seeds. Well, when you actually go back and look at the facts, yields among those who used biotech seeds, cotton seeds, yields were up by 25%. Individual farmer profits by an average of 50%, and living standards by 18%. By every economic measure, the data show that this is beneficial. And among those who adopted biotechnology, suicides actually have dropped. So I won't get into that, but glad to add more to it if there are questions about it. And there are now many well-done studies looking at the impacts of GM crop adoption by modified traits, and this data supports the previous slide. So overall, increase in productivity, increase in economic return, decrease in the use of pesticides, and for the environment, reduced CO2 emissions because you're no longer tilling the ground and using other methods that increase the release of CO2 into the atmosphere. So this is a summary of the megatrends that are shaping agriculture today, the need for increasing protein and food to support a growing world population. And let's come back to why this matters to children. And I'm going to try to tie together income, availability of food security, and also drill down on a couple of specific nutrients. Iron, vitamin A, and zinc are among the top 10 nutrition-based risk factors for global burden of disease, according to the WHO. Every one of you knows that vitamin A deficiency has some very significant consequences. 1 to 3 million children each year die as a result of vitamin A deficiency associated with concurrent measles or diarrheal disease, and up to 500,000 children still go blind each year because they don't have adequate vitamin A. We know how to provide vitamin A. It can be given as a supplement in drops or sprinkles, tablet form, but that's expensive. It requires distribution systems, and it requires compliance. Well, here's a novel idea. What if you could take the staple food that these children and families are eating and enhance the amount of vitamin A in that food? Well, it turns out that we actually know the pathway for vitamin A production, and it depends on 6 enzymes that convert precursors into carotenoids, which in our body is then turned into vitamin A. And so about 25 years ago, this fellow on the left, Ingo Piotris from Switzerland, together with a colleague Peter Bayer, took 2 of those genes and put them into corn and found that they could enhance the level of beta-carotene in the corn, and coincidentally, they didn't anticipate this, they could also enhance the level of iron in the corn. So we're hitting 2 of those 3 critical nutrients with this genetic modification. Now, this was a proof of principle study. And so what they went on to do was to, instead of taking those genes from a daffodil, they took it from a specific breed of corn that produces more vitamin A. And with those 2 specific genes, they were able to increase the amount of carotenoids by 90 to 100-fold. And they showed that daily consumption of normal quantities of rice with this boosted carotene was enough to support the daily needs of children and adults. And then a study was just published a couple years ago. Bob Russell at Tufts and a group of colleagues that he was working with in China showed that, in fact, consumption of golden rice was sufficient, it was biologically available to the children. They had vitamin A levels in their blood that were similar to those children who got vitamin A as drops or in oil. And in fact, others have done this with other staple foods. Canola oil, for example, in 2000. And I want to highlight the fact that this is 2000, so that's 15 years ago. But golden rice and golden canola oil are still not commercially available. And they're not commercially available not because the science hasn't demonstrated that they're of high value, or because the products aren't available, or because they're too expensive. These are genetically modified traits produced by nonprofit organizations that have agreed to make the seeds free, available for free to farmers to plant. Why are they not available? They're not available because largely the political environment really won't permit that to happen. And here's some of the hysteria that we see in the blogosphere and other places talking about this. And Greenpeace, of all organizations, burned fields where golden rice was growing in the Philippines. Can you imagine burning rice fields in a country that desperately needs the rice to feed its population? So I'm cruising into the end of the talk. I know I've exceeded my time, but I just have a couple more slides. I hope I've begun to make a case to show you that biotech crops, together with modern farming technology, can help to address global food issues. That it's available to and can help the small farmer. It can spur economic growth, reduce the use of chemicals, increase yields, increase the income of poorer farmers, and provide that security that allows them to have adequate food to support the needs of their growing children. It's sustainable, it's safe, it can improve nutrition and health, and it complements conventional and organic farming. There's nothing exclusive about the use of biotechnology together with conventional breeding and conventional farming. Golden rice is a perfect example of that. The initial strains of golden rice that were produced in the laboratory were not in strains of rice that are commonly available, or consumed, I should say, in certain parts of the world. And so what they did was to crossbreed, perfectly conventional, crossbreed the golden rice with the rice, the species, the varieties of rice being grown and consumed in these particular areas, so that the people would have available to them the conventional rice that they usually eat, but with that new added trait. I love this particular blog. It's from a mom who is also a farm wife from Western Canada. She's also a nurse, and I think she's made a very bold statement here. So these are my concluding slides. I started with this slide. I said this is an important reason for us to pay attention to this subject. So what about glyphosate? To repeat, it acts on an amino acid pathway that is present in plants. It is not present in any animal or human being. It is completely water soluble, so it doesn't accumulate in fat, and therefore it isn't stored in the body. It's largely passed in the stool, the minute amounts that may be consumed, and what isn't eliminated in stool is completely eliminated in urine. And there's now a 30-year history of safe use, and this was most recently assessed by the European Union through their rapporteur agency, the German Agency for Risk Assessment, and they looked at 400 publications produced over the past 40 years and concluded that this was perfectly safe. The EPS and the FDA have repeatedly examined this, most recently 2 years ago, and found it to be safe. As a result of the report by the agency of the WHO, and by the way, 3 other agencies of the WHO have found that glyphosate is safe. This one that was reported last April or May raised the issue that there was a risk of it as a carcinogen. Every other agency of the WHO has confirmed that it's safe. The EPS is going to do another review of this. They're in the process of doing another, yet a 3rd review of this. I think there's no doubt that it's going to come out as safe. There was a study of 90,000 farmers in North Carolina and Iowa, supported by the National Cancer Institute, done over a 20-year period. And they specifically looked at cancer incidence among those who use glyphosate, both incidentally, just incidental consumers, as well as those who live near farms and who live on farms. And they found no increased incidence in cancer in this 90,000 cohort. So what then why has there been a fuss about this in the pediatric community? Well, this came out over the summer. It was a so-called study of 10 mothers, breast milk, sponsored through Moms Across America and Sustainable Pulse. This is two anti-GMO organizations, and they sent breast milk and urine samples from mothers to one of these groups that does assessments of chemicals in body fluids. Only the method that they use to measure glyphosate is a method that's used to measure residues on the top of plants. It's not a method that's used to measure residues in complex biological fluids that have fats and proteins and sugars in them. And so this is what they reported. It led to the kind of blog in the previous slide. Fortunately, over the summer, a paper was reported that showed absolutely no risk and no glyphosate in any breast milk sample. This was a study sponsored by the NRSA, so sponsored by the U.S. government, conducted in Idaho, Moscow, Idaho, and Pullman, Washington, and 41 women. And again, they looked at some of the women who lived near a farm or adjacent to a farm, some with conventional diets, assessed milk and urine, and they used an assay that Monsanto has been using because they have very sensitive assays for this and have been doing this in animal studies for years. And then they looked at it in a completely independent agency called Covance Laboratories in Wisconsin, which has nothing to do with Monsanto or any other agriculture company. And what they showed was that using highly sophisticated technology, there is no glyphosate in these samples. And where it was present in urine, it was either buried, when it was detectable in urine, it was barely detectable and at levels 1,000 times lower than is permitted by the government. So I have a list of references if you'd like to look at this for yourselves or for patients. And I'm going to conclude with this slide. I won't eat anything that's genetically modified. It could be unhealthy. And I always end every talk with a slide of 2 of my 3 granddaughters. And this is outside of our house in Maine. And the 3rd one is now just 3 weeks old, so she's not yet enjoying the beach, but she'll be in this picture next year. Thank you very much.
Jose Rodriguez, pediatrician here, Marietta Wellstar. The probiotic has taken the imagination of everybody, science, non-science. Could you address briefly maybe prebiotic?
Prebiotics.
Yeah, the fibers and the good stuff we're supposed to.
More Dr. Kleinman's.
Well, prebiotics are what feed probiotics. And so largely we've been looking, or those who are in the field are looking at oligosaccharides. Those substances actually, which are fairly rich in human milk, turn out to be very useful in growing the Bacteroides and Bifidobacter that are considered probiotics. Yes, they are. And in fact, many infant formulas now have galacto or glucose oligosaccharides in them. There are probably 20 or 30 different varieties of oligosaccharides that are available now. Well, in theory they can. Any food that contains oligosaccharides can serve as a prebiotic. I don't think we really fully understand the diet mix that's best going to support a probiotic ecosystem in the gut. And it's also not entirely clear, at least to me, what a probiotic ecosystem is. I get a little anxious when I hear people, and Jeff, you should really be the person to comment on this, but I get a little anxious when I hear people classify bacteria as good bacteria or bad bacteria among the gut bacteria. For example, Pseudomonas, which in most, if we had some more lotto tickets and I asked how many people consider Pseudomonas a good bacteria, you'd all be sitting on your hands. On the other hand, colonization with Pseudomonas at an early age in animal studies shows that that leads to a state of immune tolerance in gut immunocytes. So gut bacteria each have their distinct roles. Those roles play out in the specific ecosystems within the gut, and they play out at specific times of human development. And all of this is something we're really learning about. And so I think it's very premature to understand how you can best feed the gut to produce optimal environment down there.
I think it's incredible the amount of probiotics that are available, and there's no regulation. You don't know what you're getting, what they're contaminated with, whether they really are alive or not. But we use a lot of Culturelle in kids and diarrhea and C. diff. Well, there was a Culturelle study in kids with Crohn's disease, and the Data Safety Board terminated it early because those children were doing worse. The ones on the Culturelle versus placebo were actually doing worse. So we really don't know at this point what bacteria to take to spur health and certainly not to treat certain conditions.
I think we're right to have a concern that promiscuous use of antibiotics has consequences. Principally antibiotic resistance and then inability to treat common infections over time. Increasingly, the producers, particularly animal, chicken, beef, pork, have been reducing the use of antibiotics in growing these animals. It was probably 30 years ago or 40 years ago that it was discovered that if you give antibiotics to animals, that it actually enhances their growth for reasons that aren't clear. So if you're a farmer and you're trying to produce a product faster and a bigger product, meaning a chicken, let's say, you treat with antibiotics and you get a bigger chicken faster. But all of this has begun to be balanced by a recognition of what that does, and so there's been a dramatic effort to reduce antibiotics. Milk is one area where we don't have to worry as much because milk is tested for antibiotics, and if residues of antibiotics are found in milk and there's a panel that are tested, then they have to discard the milk. It's not allowed into the marketplace. So I think that's one food where antibiotics haven't played a role recently.
I have two questions. Number one is that can you please comment on the fear of the monopoly, like companies like Monsanto, in terms of selling the seeds to the farmers? And the second question is, you said we don't know what the effects are in the future of this genetically modified products. So what do you think should we look for?
I didn't get the second question. I'll speak to the first and then let's go to the second again. The question was whether or not we're at risk of being totally dependent on a few large companies for our seed supplies. In other words, what's the risk of a monopoly on seeds by companies like Monsanto? Over time, the number of companies that are producing seeds has grown dramatically, but it's a very expensive process to produce a genetically modified product. The same, however, is true for conventional products. The fact is that all farmers who sell their fruits or vegetables purchase seed year after year. If you harvest your own seed, if you just reuse the seed from the year before, take a tomato, whatever you don't sell, you use the seeds from the tomato and use the next. They're not very stable, and they don't produce the same quality product year after year. So the fact is that farmers who depend on farming for a living almost all buy seed year after year, and they buy seed from just a couple of companies. Organic seeds, for example, the largest producer of organic seeds is Mars. The candy company, they have a subsidiary company, and I'm blocking on the name of it now, that sells seed to most organic farmers. So the short answer to your question is that there are multiple companies making seeds. They're dominated by about 5 big companies, but there are many smaller companies. For example, the Arctic apple that just came on the market that doesn't brown as fast and has a longer shelf life. That's produced, that was derived by a very small company in Western Canada. So I'm not particularly worried about that any more than I'm worried that GM, Ford, and Chrysler have a monopoly on cars. I think the marketplace is pretty good at sorting out this issue of monopoly, and I don't see any danger that we're moving in that direction. Now, what was the second question?
The second question was that you were saying that we do not know in the future what the effects could be of this genetically modified products that we are eating. So what do you think should we look for?
Yeah, I think we actually do know that genetically modified products are safe, and through the testing that's done on these, compared to conventional products, they're at least as safe and probably safer than conventional products. I don't think we have anything to be concerned about in the future from the genetically modified fruits and vegetables and animals that we're eating today. I do believe in testing. I think it's very important, and I think it's a very good thing that extensive testing is done on these products. But I would like to see the same kind of testing done on other ways of producing seed. I mentioned mutagenesis and radiation. I think if we're going to test anything, those are the products that ought to be tested. But even without testing, there hasn't been any adverse effects. And in the 30 years that we've been consuming genetically modified using insertion technology, so single or several genes, there hasn't been a single reported confirmed adverse effect of a genetically modified food. That's over 30 years. So again, I think they're perfectly safe. I have no qualms in eating them myself, giving them to my grandchildren, or recommending to others the same advice.